Hello. Today we're going to discuss Edexcel Biology Unit 4 questions. I have selected this question as a first question. Explain why the use of an enzyme to cut the DNA results in fragments of different lengths that can be separated by gel electrophoresis. This question carries 3 marks. First of all, students should analyze the question before they're going to write the answer to get the 3 marks, maximum marks for this question. I will show you first how you're going to analyze this question to find the exact points that you need to write to get the 3 marks. The first, this is explain type of question, and for explain type of question you have to show the mechanism, you have to show the exact process of how this take place. Use of enzyme to cut the DNA. That means if enzyme used to cut the DNA, it can make fragments. Why enzyme make fragments? So first thing, when you analyze, you have to answer this question: why enzymes make DNA fragments? The second thing, why these fragments are in different size or length? The fragments are in different length. Third one, this can be separated by gel electrophoresis. So different length can be separated by gel electrophoresis. So now it is clear your 3 points: why enzyme make fragments, why enzyme make different length of fragments, why different lengths can be separated by gel electrophoresis. So if you can provide answer for these 3 questions, you can get a 3 marks. Okay, let's see again. Enzyme can make DNA into different fragments. How? So we address that question first, right? This represent DNA molecules, double-stranded DNA molecules, and you can see 2 regions. One is yellow region, other one is a red color region. If you recall your knowledge on DNA molecule, at AS level, you have learned DNA molecule has 2 regions, exon and introns. So when you are advancing in biology, you can learn many regions, different regions in DNA molecules. So I am going to introduce another important region we can find in the DNA molecules. Because initially when we learn DNA molecule, we have an impression the DNA molecule consists of genes and the genes make proteins. With that, we think DNA molecule from one end to another end, uh, genes are located. But that's not the correct, because we try to understand around 1.5 to 3% maximum covers the gene parts of the DNA. So now more than 97% represent other regions of the DNA molecule. So I'm going to introduce another different region, this red color areas. Here one, here one, here one. And these red color regions are called restriction site. Restriction site. There is another name, but we don't use in Edexcel this name, which is called palindrome sequence. The restriction site has palindrome sequence. So I will not too much going detail to the palindrome sequence. I use just to avoid the complication restriction site. Alright, now here you can see one restriction site, second restriction site, and third restriction site. So what is the speciality of this restriction site? their location is not very precise. The length is not very precise. It's a randomly located. So if we take from here to here, there is a 1. And from here to here, there is a 1. And here to here, there is a 1. So the location of these restriction sites are different. They are located at different position of the DNA molecule. And what is the function of the restriction site? These restriction site are the specific area for restriction endonucleases. Restriction endonucleases. Restriction endonucleases are enzymes. And these enzymes are specific for what? If you learn enzyme active site are specific to the substrate. If we consider DNA molecule is the substrate here, restriction enzymes are the enzymes. So this specificity is different in this case. Specific for sequence. Restriction endonucleases are specific for the sequence. So restriction site has a specific sequence. So what is the specific sequence? Example, if I write from 3 prime to 5 prime, AACGTT. And on the 3 prime to 5 prime, this direction, I can write AACGTT. Now, if you look carefully this sequence, the nucleotide from 3 prime to 5 prime, AACGTT, and the reverse sequence, AACGTT, the same sequence. So these sites are called restriction site, and this is the specificity, the sequence. So what will happen now if we use restriction enzymes to cut this DNA? This can cut DNA into 1, 2, 3, 4 fragments. Being restriction sites located at different places, the lengths are different. Right, next, if we place this on load to the gel, their sizes are different. The DNA molecules are negatively charged. They are moving from cathode to anode in the gel electrophoresis. This distance of traveling in the gel depend on the size of the DNA molecule. Larger fragments move less. Smaller fragments move far. So look at these 4 fragments now. So they're going to move like this. The smaller fragments move faster. This resulted a different position in this banding position in the gel electrophoresis. Okay, so if we analyze the gel now, we can see the large band here, and then the second large band is here, and the third one is here, and the smallest band is this. Right now, if you go back to the original question, why the use of an enzyme cut the DNA resulting fragments? Because there are many different restriction sites, and their locations are different. And this resulted different size of DNA molecule. As a result, they separate during the gel electrophoresis. So when you want to write answer, this is the correct answer to get the 3 marks. This is because restriction enzymes recognize specific base sequence of the DNA. These recognition sites are not equally located along the DNA. The restriction enzyme hydrolyzes the phosphodiester bond of the DNA. Therefore, due to different size of the fragment, DNA move different distance through the gel. When you write the answer, you should try to write answer like this to get the full marks. Okay, then we try to go to the next question.